with that being said, thank you for today's Water at Wayne. It's my distinct pleasure to welcome Dr. Krista Wigginton from the University of Michigan. So she's here today to talk about how environmental engineers and scientists have been helping to address the COVID-19 pandemic. And this is a great person to have here, since this is her research has focused on coronaviruses even before the pandemic. So Dr. Wigginton received her bachelor's degree from the University of Idaho, and then went to Virginia Tech, where she received her master's and PhD. She then spent some time at EPFL in Switzerland, and she's currently an associate professor at the University of Michigan in Ann Arbor, professor of environmental engineering. Her research team focuses on viruses in the environment, the mechanics, the fate and transport, and how these, what role they play in urban water microbial ecology. She's also done quite a bit of method development, and I think you'll probably hear about some of that today. She's the recipient of an NSF Career Award, and of course that Career Award was focused on envelope viruses and wastewater, so perfect timing for that award before a global pandemic in which you had coronavirus become a problem. She's also received the Water Research Foundation's prestigious Paul Butch Award. And with that, I don't want to continue on and take up all her time talking about her accomplishments, because there are many. But with that, I'll welcome Krista to the floor here. Thanks, Krista.
Yes, thank you, Sean. Thank you so much for having me, and thanks everybody for listening today. Wish I could be in person. Remember, I traveled, the long trip to Wayne State several years ago to visit Sean when we were both assistant professors, and would love to come back and talk in person one of these days, hopefully soon. So let's see, I'll get right to it so we make sure and have time for discussion at the end. I do, I notoriously I'm hurried at the end, so I want to make sure and acknowledge, acknowledge those who should be acknowledged early on. My research team here on the left, this is out at Pinckney, and then the funding sources for some of the research I'll discuss today. And then there's quite a few collaborators that I definitely want to, I know I'm not going to get to give them as much, I'm not going to be able to highlight their work as much as I'd like, but everyone here has really contributed to the work that I'll present. So with that, I think it's kind of nice to think back to the early days of the pandemic, or back then it was just an outbreak, and think about what we were worried about, what we didn't know, how scary it was. I mean, I remember, I'm sure you all recall leaving things outside for a week before bringing them into your apartment, going for walks and worried about passing people on the sidewalk, water, food. It was just all over the place. We had ideas about how we were getting, people were getting COVID, but there was still a lot of unknown. And so during that time, I feel like environmental engineers and environmental scientists really stepped out to help out the other critical fields in helping to answer some of the questions we had. And then once we learned where the viruses were at and how they were being transmitted, to use our skills to help address the pandemic. And so I'm gonna, and when I made the title for this presentation, you know how it is, it's months ahead of when you're gonna present, and I went bold. I was, let's talk about what have we done for this pandemic? But then when it comes to actually making the presentation, that's pretty broad. So I'm gonna narrow it down a little bit here and focus on 3 ways that I feel like our field has really contributed. So when we think about our toolkit that we had coming into this pandemic, some of the things that were relevant were, we were very good, our field was good at characterizing viruses in the environment. So where would they, if they're released into water, where do they end up in water? How are they inactivated in water? Same thing with air, if they're released into air or onto surfaces. We're good at that, and that's what we do. And then once we find out they're there, our field is excellent at taking care of them. So getting rid of them, disinfecting water, filtering water, using UV treatment on water, air, and surfaces. So that's another place where we had a role to play here. And then I think, typically with detection, we do a lot of detection. Our field does. Usually we're thinking about it in terms of what are the risks being posed. But in this pandemic, we've had a new opportunity to use detection with surveillance. So I'm going to show, some of the things I'm going to talk about in these realms today are the work that's happened in our field. I'm going to touch on it briefly. This is not my area, but just want to definitely give credit to some key players that have really helped to define and communicate how aerosols are playing a role in COVID transmission. And then with disinfection, our field really stepped up to help with decontaminating personal protective equipment, other items that we were concerned about in the early days and throughout the pandemic. And then with the detection, I'll definitely spend probably the most of my talk on surveillance, because that's where most of my effort, my time in the last year has been focused. So a couple of things I want to just put out there right at the beginning to make it clear. We're talking about an enveloped virus here. It's a SARS-CoV-2 is enveloped. It has a single strand RNA genome. It's a big single strand RNA genome, much bigger than a lot of the other single strand RNA viruses that we care about. In water, we don't usually think about envelope viruses. When we're thinking about waterborne disease, foodborne diseases, those are typically non-enveloped. So what that means is you've got a little genome, it's wrapped up in a protein coat. But then these envelope viruses have a lipid bilayer that they get from their host cell, kind of encompassing that protein coat. And so that sets them apart. And so in this talk today, I'll keep referring to envelope, non-envelope, because that's really a distinction that we need to think about with SARS-CoV-2. Another thing I just want to mention is how we measure viruses, how we detect them. For some of you out there, I'm sure this, but for those that don't, we can do either culture-based or non-culture-based. Culture-based is like what it sounds like. You're going to culture the viruses, but viruses on their own won't replicate. You need to have the host cell for them to replicate within. And so we need to have a host cell system for a specific virus that those viruses can infect and replicate in. And then once that happens, we see different effects in the host cells. We'll see plaques form. That's what I'm showing down here on the circle here. That's a lawn of cells where the viruses have created plaques. We can use immunofluorescence microscopy and other methods to see whether those cells have been infected and quantify how many infectious viruses we have. Now, this type of method to quantify viruses, either if we want to go out into the environment and quantify viruses, or say we're working in the lab and we want to test a disinfection strategy, we'll throw viruses in, we'll culture them before, we'll culture them after, count how many were there. We're relying here on a cell system that can be infected by the viruses, and that there are a surprising number of human viruses where we don't have these cell systems, or maybe working with these viruses requires really high levels of biosafety that most labs don't have access to. And so in cases like that, we're relying on molecular methods, where here in the red, I'm showing a little piece of maybe the SARS-CoV-2 genome, it's the RNA. We'll use this reverse transcriptase enzyme, make a copy of DNA, and then use PCR to make lots of copies of that DNA, and at some point quantify the DNA. But then we can back out how many copies of the original RNA genome we had present. So we rely on this a lot for SARS-CoV-2 environmental surveillance. We're not going out typically and trying to culture the viruses. We think, in fact, they're not infective in most cases. So these molecular tools are targeting a specific region of a virus, and we've designed them so we know it's the virus that we're looking for. So we got qPCR and culture-based methods for this envelope virus. And so the last thing I want to mention before I get into more of the details on what our field has done, I want to mention that we rely a lot on surrogate viruses. We rely on, so these are viruses that don't have the same pathogen, they're not as pathogenic as SARS-CoV-2 or Ebola or even norovirus. We often use bacteriophages as surrogates. So we can grow those in bacterial cells, and they have the same components as human viruses, but they're easier to work with. We can grow them to high quantities, and we can test them very quickly. And so usually what we'll do is we'll take some bacteriophages, maybe an animal virus that's similar to the virus we're looking at. We want something that we want them to have characteristics similar to the human virus that we're looking at. So I'll talk about a lot of surrogates through this discussion that our group and other groups have used to try to pull out what's happening with SARS-CoV-2. All right, so first, going back to those kind of 3 things in our toolkit and the way we've applied them as a field, I definitely want to talk about aerosols and transmission, because this is a virus that, it's not a virus where we're concerned so much about water or food. This is a virus that's transmitted through aerosols and droplets. And so I've got to give some credit to Lindsay Marr's team at Virginia Tech and many others. I should mention here, I'm not coming close to capturing the whole field, so I'm just pulling out a few people here and there and a few examples. But Lindsay Marr's been working on, viruses and aerosols and droplets for many years now. This was a paper that was actually published, I think it was submitted just before COVID came to light. And so what they were looking at here is how relative humidity impacted the survival of viruses in aerosols. And she was looking at 2 different surrogates here. These are those surrogate viruses. We have Phi6 is a surrogate enveloped virus, and MS2 is a surrogate non-enveloped virus. They were looking at how humidity impacted the persistence. And so they're looking at the relative viability. So the more of a drop they see here, the more inactivation that they're happening. And so they're seeing that relative humidity in the mid range led to more inactivation, where lower ranges and higher ranges for both of these viruses led to less inactivation. They also studied how different humidity conditions led to the formation of aerosols, and looked at how the solute concentrations in those aerosols impacted the infectivity of the virus. So really cool work that was critical coming into SARS-CoV-2. And Lindsay and others in this area who are environmental engineers and environmental scientists have not only done some really cool work over the last year, but they've also, been out there really communicating aerosol science and the role that aerosols are playing, and really trying to convince the big the bodies that come out and make statements about how it's being transmitted and what people should do, really making a case for aerosols in the transmission of SARS-CoV-2. And so I just wanted to definitely say that's one area where our field stepped up and has been playing a really important role. Let's see, so then I'll go on next to our next piece of our toolkit, the disinfection or decontaminating surfaces and water. And so let's see, so we knew going into this a little bit about coronaviruses and envelope viruses. Like Sean mentioned, my team had worked with envelope viruses and looked at kind of different conditions in which they're inactivated and which conditions are persistent, always kind of comparing them to non-envelope viruses. So this data here is for free chlorine. We looked at an envelope virus here on the left. This is Phi6, a surrogate envelope virus, and a non-envelope virus here on the right, MS2. And what I'm showing here is the free chlorine inactivation rate constant for these 2 viruses. And we can see that the envelope virus is much more susceptible to inactivation. The envelope virus is much more susceptible to inactivation than the non-envelope virus. And we did similar work with wastewater. We looked at envelope viruses in untreated wastewater and non-envelope viruses in untreated wastewater and compared those inactivation kinetics. And similarly, we saw that the 2 surrogate envelope viruses are much more susceptible to inactivation in untreated wastewater. So in solution, we kind of knew going into this pandemic that this is an envelope virus. If it is released into water, it's going to lose its infectivity quite a bit faster than the non-envelope virus, and it's going to be more susceptible to our typical drinking water or wastewater disinfection methods. And I should say we're not the only team that looked at this. Mark Sobsey's group, Gerba's group at Arizona, they've also looked at envelope viruses and coronaviruses in particular and shown that they do tend to be more susceptible. So we knew kind of that water wasn't going to be a driver here. But that didn't mean that surfaces weren't important. And so what did we know about coronaviruses and envelope viruses, I guess, coming into the pandemic when it came to surfaces? And this is some work by Mark Sobsey's group. Lisa Casanova's done quite a bit with envelope viruses. They looked at what happened when these are 2 different, each of these different colored markers here are 2 different coronaviruses. They're animal coronaviruses, so not human coronaviruses, but very similar to human coronaviruses. And they looked at different temperatures and relative humidities if you did have the coronaviruses on surfaces. the main take home of this early work, this was from 2010, was that higher temperatures have higher inactivation kinetics for the coronaviruses, and that higher relative humidities also resulted in higher inactivation rate constants. And so going in, we knew that if it was cooler with low relative humidity, these viruses were probably going to persist longer. And so this was all work that had been done before, but now coming into the early days of the pandemic, you probably all are very familiar with the global shortages of personal protective equipment. So the N95 masks in particular, we were scrambling in those early days to figure out how we were going to stretch these really important resources out so that we could protect our medical professionals. And so I want to mention this interesting paper that came out of ES&T Letters in 2020 out of Illinois by some environmental engineers and scientists, where they designed a method using electric cookers, so like rice cookers, where you could take your own N95, if you were a healthcare worker, take your own N95 and heat it in a rice cooker. They were using dry heat. What is shown off to the right here, this is the inactivation of the virus, the log inactivation of the virus, different viruses that they looked at on these, that had been deposited on these masks over time. And this is at 100 degrees Celsius. And so at the end of this paper, they recommended, I think, 50 minutes at 100 degrees of dry heat was effective with, one at a time in an electric cooker. Our group also did some work on N95 masks. We worked closely with Michigan Medicine early on in the pandemic, and it was intense. They were trying to get a large scale reprocessing method down in a short period of time where they could expand it across the whole hospital. And so that trying to do it at that scale limited the options that we had, right? We needed something that we could go through hundreds of masks a day. And so we looked at, like many places, I think a lot of us were doing this in parallel as fast as we could and talking and exchanging ideas and experiences. We tried heat, we tried UV, and hydrogen peroxide vapor. Those were the three main processes that we attempted due to their ability to scale up for the hospital. We landed on humid heat just because it was the easiest to scale. It was fairly inexpensive to scale, and we've got really consistent results across different viruses. I'll show you some of the results here in a little bit. So we landed on 30 minutes because we felt like that was a reasonable amount of time to have high throughput, and we saw really good inactivation. And I'll explain why the humidity was important in our case. So what we did is we had some surrogate viruses. We put them onto the masks. We dried them on the mask. And I want to give credit to these students down in the left corner and others too that aren't named here, but particularly these three. They worked tirelessly for the first. Like when things were shutting down, they were going into the lab day after day, night after night to crank out these experiments and really put this data out there so people knew what to do with their N95 respirators. So they put the viruses on, do the different treatments, pull the viruses off, and then plate them and see how the treatments were doing. These are the viruses we used. I talk about surrogates. There's some surrogates. MS2 is a non-envelope virus. That's the only one we used because we really were trying to understand what would happen with the coronavirus. 5.6 is a typical enveloped bacteriophage, so we can get it to really high titers. It's easy to work with. But then we also wanted to work with some animal viruses. So influenza A virus, Adam Loring helped us out quite a bit at Michigan on this project. And his lab, Will Fitzsimmons in particular, helped with the influenza. And then murine hepatitis virus is a mouse coronavirus, and we did that in our lab. It's a virus we've worked with before. So what we observed is that when we chose 72 degrees and 82 degrees, we wanted to get it warm enough that we saw really good inactivation, but not so warm that we were worried about repeated treatments and the degradation of the mask. And so these are the 4 different viruses on each of these different plots at the 2 temperatures or the 2 colors. And when you see the colors kind of disappear and become hollow symbols, that's when we've exceeded our inactivation limits. So we've got more viruses inactivated than we can measure. But main thing, we see not much inactivation down at low humidities, some inactivation, but not as much as you hit this threshold of around 20%, 30% relative humidity, and you really see a flux of much more inactivation. So humidity really seems to play a role. One thing that was interesting, I think going into the pandemic, people were using non-enveloped viruses as kind of a hardier, they expected them to persist a lot more. In fact, some of the FDA requests to prove that processes we're working with were with non-envelope viruses to really make sure that we were doing what we wanted on SARS-CoV-2, the envelope virus. But what we're looking at here is the same data, just plotted a little differently. So inactivation on the y-axis, the log inactivation, and the relative humidity on the x-axis. And these are our 4 different viruses run in duplicate. And we didn't really see any trends between the different viruses where we didn't see that the non-enveloped MS2 was repeatedly inactivated more than the envelope viruses, which suggests that these non-envelope viruses for surface decontamination aren't necessarily hardier and aren't necessarily a conservative surrogate for the envelope viruses. I should mention that, again, this is for 30-minute treatment at these humidities, and you can see the trend, and as you go up in humidity, you have more inactivation, and the arrows show that we've exceeded our assay detection limits. One other thing to note is that these early recommendations by the USDA, the FDA, the US FDA, was that they did want to see greater than 6 log inactivation of 3 non-envelope viruses. So again, getting at that assumption that the non-envelope viruses are the more resistant in these scenarios. One thing we noticed early on in these experiments was we did one control where we put a virus in a couple of different solutions and we saw wildly different results. And so after we kind of finished this large scale, work for Michigan Medicine and kind of got their process going, we wanted to go back and probe what was going on with the different solutions and could that be biasing results. So we took these 2 phage that, again, we can measure up to really high levels. That's why we like to work with them. We can crank through the experiments, and we showed that they behaved similarly to the coronaviruses and the influenza virus. So we took them and we put them in different media, and then we deposited them on the mask and let them dry, and then looked at how they were inactivated through heat treatment. And what we saw really surprised us. So we put them into PBS, so just a buffer, into the media that we use to grow the cells and the virus, and human saliva as kind of what we would expect maybe them to be on a mask. And then the PBS buffer with some protein added to it, because some people had noticed that BSA or protein might affect the persistence. What we saw is that when the viruses were deposited in the culture medium, we had much more inactivation across the board for the viruses at different conditions, regardless of the relative humidity or the temperature. We saw this again and again. And that it might seem like a small, who cares about this effect with media, but it's really important because a lot of studies, because it's so hard with human viruses to pull them out of the media that they've been grown in and put them into something that's more, representative of what would be actually the virus from a human would be present in. It's really hard to do that. And you lose virus and you're starting with such precious virus to begin with. People usually just leave it in the media and do experiments on persistence in the media. But this suggests that we would measure way more inactivation in the media. Oh dear. the crash just happened. So give me just a second. I will open this back up. Did not like that slide. Here we go. Got it open. Go back to the slide. Share my screen again. And go to the slide. I'm going to swap displays. Okay, I think this should be right. Please speak up if you're not seeing what you should be seeing. So the bottom line, the studies that deposit viruses in the media that they were grown could be overestimating the inactivation or the decontamination that's actually happening with the human viruses in saliva. And so we recommended after this study that for future studies to really, really think about the media that you're depositing the viruses on, whether it's for a surface decontamination, masks, because you could be having this real enhanced inactivation that's not going to happen in saliva or whatever other bodily fluid that the virus is in. Okay, and then I'm going to spend the last 15 or 20 minutes talking about the environmental surveillance, which is really, really exciting work that's coming out of so many labs right now and helping out quite a bit with the pandemic. So if we rewind back to SARS-1 in the early 2000s, this is an envelope virus. It's not the type of virus that you would typically go looking for in feces and wastewater. But these are pictures. This is a picture of the Amoy Gardens in Hong Kong. In this instance, they did think that feces played a role in the transmission between cases in this apartment complex. And sure enough, when they went and looked for SARS-CoV-1 in feces, they did detect it in quite a few feces samples, fecal samples. And so this was a paper from 2012 from a group in the Netherlands. And I hadn't read this until recently. And I read this last couple paragraphs of their paper. And I was just like, wow, they were ahead of the game. This was a paper that they'd written on poliovirus surveillance with wastewater, something that has been going on for some decades. But they noted that what they were doing in the study was trying to correlate the amount that was in wastewater of polio with the number of cases. They've done some challenge testing in the community where they actually infected people or I think vaccinated people with polio and looked to see how much they could detect in the wastewater. So they were calling out, back then that this would be a really helpful surveillance tool if we had another SARS-like coronavirus come along. And then sure enough, another group in the Netherlands, I don't know if this is a coincidence, I've never asked, but was one of the first groups to start looking for SARS-CoV-2 early last year when we had the pandemic. So an early paper in ESNT where they looked at the prevalence of COVID in the community, measured it in the influent at the wastewater treatment plants, and showed some nice correlations between the levels in wastewater and the amount in the community. So this has really exploded, right? I mean, I know you all have heard about it. I'm sure some of you in the audience might even be participating in some of the Michigan projects on this. But so this is a preprint that was just put up, I think, last week or the week before from Colleen Naughton's group and a bunch of others who were doing work in this area, where they tried to kind of track how much SARS-CoV-2 wastewater surveillance is happening around the globe. And bottom line is there's a lot, and a lot in the United States. Hundreds of groups in the US are doing wastewater surveillance. So there's something to this. It's not just a flash in the pan. It's going to be around, I think, beyond SARS-CoV-2. So a little bit on our work in this area. We had studied, as Sean mentioned, we'd studied coronaviruses in wastewater. Yin Yi, who's now an assistant professor at University at Buffalo, was a PhD student in my group. And she spiked in these surrogates. Again, we've got a coronavirus and enveloped bacteriophage, and then the two non-enveloped surrogates that we work with. She spiked them into wastewater, looked at how they survive and how they absorb, and how she could best recover them with different methods. And one of the things she observed, this data is looking at the partitioning and the inactivation of the non-enveloped virus MS2 and the enveloped coronavirus, the two of the viruses we looked at. She noticed that for the two non-enveloped viruses, we had less sorption to the solids and influent. So she took influent that hadn't been settled yet and spiked the viruses in. There was less partitioning to the non-envelope viruses. So more sorption to the solids from the envelope viruses. And if we use the data from that paper to kind of calculate the partition coefficients for the two non-envelope viruses and the two envelope viruses, we saw about a 10x difference in the partition coefficient. So, and coming out of that, we were not thinking about how could we use this for surveillance. It was more about if there was ever a pandemic and we were worried about viruses in wastewater that were infective, where would we be most worried about them being at the wastewater treatment plant? So jump ahead to last year, I was on sabbatical at Stanford working with Ali Baim. And we, thought, like many others did early in the pandemic, while we were just kind of waiting for the first cases to come to California, what if we started measuring wastewater right away? And maybe we could catch it coming in California before any cases were ever even reported in the United States. Turns out there had already been cases in the United States. We just didn't know it, but we started this project anyway, got an NSF rapid grant, followed by some private funding afterwards. And so because of that work with Wigginton and the focus on solids, we decided to look at both solids and liquids. So looking at both settled solids at the wastewater treatment plant and at the influent at the wastewater treatment plant. Kind of sets our work apart from a lot of the other groups. And so why did we focus on solids? Well, I mentioned before, the coronavirus partitioned more to solids than the non-envelope viruses. Both of the envelope viruses did. But, and so we early on, we started sampling both solids and influent, and I'll show you some of those results in a little bit. But if you think about the partitioning, some groups have argued against solids because what they'll do is they'll take, say, a liter of influent, and they'll collect the solids from that influent and measure what was in the influent and in the solids. And so based on this KI, and they won't detect much difference when they remove the solids from the influent than if they left it in. And it makes sense because with this partition coefficient, if you take, say, influent and it has 250 milligrams per liter settlable solids, and you have a total of 10 viruses in that liter, then most of the viruses are going to be in the liquid fraction or associated with the liquid fraction. Only 2 would be associated with the 250 milligrams of solids. So if you're only looking at those solids, it wouldn't make sense to focus there rather than the larger volume of water, because there's so much more water than the solids. But if you go to a wastewater treatment plant, you have this automatic enrichment process going on. You have primary sedimentation where you settle out the solids. And so, if you think about collecting from the influent, where you might collect a liter versus some solids, some sludge coming off, or I should say settled solids, I wanted to kind of walk through how this might look. So if you have settled solids, say it's 40,000 milligrams per liter, and I should say, I need to check my math. I did this pretty quickly, but we can, I'll correct it and let you know if it wasn't quite right. I think that the general idea holds. So if you settle out the solids and then take, say, 50 mils, say a conical tube of those solids, then based on that KID, what you've done is you've enriched the viruses in that volume, and now you've got 16 viruses associated with solids in that settled solids, and only 0.5 is what I calculated in the liquid associated with the liquid fraction. So you can see how we need a liter of influent to get 10 viruses, where here we need 50 mils of solids to get 16 viruses. So it's like you've enriched the sample for the viruses. And then what we've done in our group is, rather than just extract directly from the settled solids, we'll settle it more, we'll centrifuge it, we obtain a pellet that's about 25% solids. And so then you're talking about, say, an 8-gram pellet that's 25% solids, that's got all 16 of those viruses in it. And the main thing here is that it's easier this way because you don't have to start with a liter or a half liter of water and concentrate it all down with multiple steps. People use PEG precipitation, ultrafiltration. All we have to do here is take solids that have been settled, centrifuge them for 30 minutes, and then do a direct extraction from those solids in the end. And so we felt like the promise of solids was that it requires sampling less at the wastewater treatment plant. You can do a direct extraction, so it's easier for scale up versus the multiple steps that happen in the influent enrichment process. And you also have fewer steps where you can lose viruses along the way. That's what we felt going into this study. And so that's why we wanted to do the solids versus influent. So we went to a couple of plants in California. This was back in February and March, and started sampling influent and solids from those plants. And we compared methods and optimized methods and tried to see whether we could detect SARS. So here's some of the data that was published in an ES&T paper from late last year. I'm showing here, these are 2 different genes, N1 and N2 in the SARS-CoV-2 genome. The top is influent samples from these plants. The bottom is, or this is one plant, solids samples from these plants where we've done in a direct extraction or a PEG-based precipitation method on top, with a pretty typical concentration factor of the influent PEG precipitation method. And the open symbols on the bottom are where we got no detects, we did both qPCR and ddPCR with similar results. On the bottom is our solids, where we have the detects and the no detects. And bottom line is, when we paired these samples, we would repeatedly get more detects in the solids than we would get in the influent. Inhibition was an issue in some of these samples, where we would have to dilute either the concentrated influent or the solids extract. And we've kind of worked out the optimum dilutions going forward. So we decided, based on that first study, we really went with solids from then on out. We kind of stopped collecting influent and got collected 50 mil conical tubes, or sometimes larger, of the solids. This is the data. What this allowed us to do is really have high resolution. We could do that every day from a plant and do a direct extraction every day. What I'm showing here this is for San Jose, California. On the bottom, this is a target in the SARS genome. On the top, these are the cases that are reported to the county. So we went daily samples, and you can see here kind of the smooth lines with the raw data for both the cases and the wastewater. We're still analyzing the gap between these on the bottom. But what it shows is that the measurements really do track nicely with when the cases are reported, like they were later on in the pandemic. And it shows us too kind of what was truly happening early in the pandemic, when reporting, clinical testing wasn't widely available. And so we can now go back to a lot of our old samples and see the dynamics early on in the pandemic. Another thing I wanted to bring up, that we've got this it was just accepted in ES&T letters. You can imagine that comparing the SARS levels from plant to plant can be tricky if you're just measuring the amount in solids, because maybe different plants have different types of solids. Maybe your recoveries are different from one type of solid to another. So there's been a lot of discussion in the field about how do we normalize our measurement or quantities of SARS genes by something else to kind of get rid of this issues with recovery and also plant to plant differences. And so what we've been doing is not only measuring SARS-CoV-2, but also pepper mild nodal virus. It's a pepper virus it's really common in our diets. And so it persists, the RNA persists through our bodies and into our excrement. And so it's present in really high concentrations in wastewater. And so we were using pepper mild nodal virus as kind of like a normalizer to address the recoveries from day to day, and also plant to plant differences. But we wanted to show, that was kind of our gut going into this, but we wanted to show or decide if there was statistics to back that up. And so what I'm showing here and what comes from this ES&T letters paper is, if we don't normalize on the left and we do normalize on the right, and if we combine data from different plants, if that improves the correlation. And the answer is, it allows us to compare data from plant to plant better if we normalize by pepper mild nodal virus. And this might be true for other normalizing factors too. Other groups use different factors to normalize, but so we're sticking with the pepper mild nodal virus for the foreseeable future. So now, I guess, getting towards the end of the talk, we've scaled up with some help from a private donor. We've got a pilot project with 8 plants across the Bay Area. Every day, a courier picks up primary solids from these plants, and it's drives them to a commercial lab where we've industrialized our solids process, and they process daily samples there. And then at the end of the day, we get the data and share it with put it on our dashboard and share it with the public health agencies. This is what our dashboard looks like, and the website's down on the right side with our daily samples from these. Right now it's 7, but it's about to be 8 plants across the Bay Area. And we've had really good success. We have a really close relationship with some of the public health agencies, and they've been really receptive to the data and are even starting to incorporate it into their own dashboards. I want to also state, we're not the only group that's doing solids. There are others. By far, it's more common to focus on the influent. I think that's partly because as environmental engineers, we're usually more interested in what's in the water than what we're getting rid of. Why do we care about what we're getting rid of? But Jordan Peksha had the same idea early on to focus on solids. His group had done quite a bit with solids in the past. And so this is their dashboard. He had a really nice Nature Biotechnology paper last year, kind of making a case for solids as well. So definitely check out his dashboard. All right, so with that, I want to again, save some time for questions. So looking ahead to post-COVID, or at least post-vaccine COVID and the next pandemic, like, where is there room for more research, and what do we need to do to be much better prepared next time? Well, there's a tendency to focus on the virus that comes out, right? When it's Ebola, we focus on how does Ebola persist in the environment. When it's SARS, how does SARS persist in the environment? But I think we need to be able to kind of take a step back and see how the new virus, we don't know what it's going to be, we don't know what its characteristics are going to be, but we need to be able to kind of better and more quickly extrapolate what we know about the viruses that we know about to this new virus. So can we predict based on physical characteristics versus biology? Can we be more confident in our predictions so that next time we're not, waiting to get the culture systems up and really kind of relying on that data on the virus to know what's happening in the environment. I think we're going to see an expansion well beyond SARS-CoV-2 and wastewater surveillance. It's a really exciting time for this. It's been around for a while, but I think it's really coming. It's becoming it'll become a much more kind of a normal thing in our day-to-day lives, that we're relying on wastewater to tell us about the health of communities and catching emerging viruses when they come up. And then I think also this is, beyond water, but for sure in a continued appreciation and increased appreciation for indoor air and the role of, ventilation and surfaces and the indoor environment for pathogens and virus transmission, thanks to our colleagues who are working in that area. So with that, I'm going to pull it back up to my acknowledgements, and I'd be happy to answer some questions.
Great. Thank you, Krista. Really interesting talk here, and we've got some questions. I'm sure there's plenty more that are about to come in. So the first question that I have from someone is, could be potentially a loaded question, so I'll let you deal with that. But why didn't we hear about this method to reuse N95s earlier? And in addition to that, is this research just coming out kind of, or were there any difficulties in trying to communicate with the media? So that's, like I said, that's a pretty loaded, there's a lot in that question, but great question.
Yeah, I mean, I do not know the answer, but I did find, when we were deep into this, of course, when it first happened and people were talking about how are we going to reuse N95s? I'm like, okay, there's got to be some literature out there on this. Like we've got to know this. And there was some, but so little. And so I don't know. I found myself asking that, how are we just now doing these experiments with different viruses and different temperatures and different humidities and on N95s? Again, there was some, but it was very, very little. People were just digging through the literature to find as much as they could. I don't know. I mean, I think there's so many different aspects of this pandemic where we were really caught off guard and we're all scratching our heads. Why didn't we know this before? Hopefully never again.
Yeah, well, while you're presenting, I was thinking to myself, I watched a lot of presentations of elected officials giving talks about coronavirus, and I was wishing that you just got to talk the whole time because I found that much more applicable. So, okay, another question from this one from Dan Schechner. Any public postings of data on COVID and wastewater in Michigan?
Oh, yes, thank you. That is a great question. And I meant to discuss that up. So, right. Yes, there is. Eagle has done a great job of mobilizing a statewide surveillance network across Michigan. We're part of it here at U of M. I focused here on the California project. But when I came back from Michigan, I got involved with Eagle and Joan Rose's group at Michigan State, and Eagle have really done a good job of equipping labs. The funding for that ran out at the end of 2020. But there is a dashboard, a really nice dashboard that shows the data that came out of that, and there is going to be a resurgence of those labs, I think, in the coming months. We just got word of that. So yes, it's been fun to be part of both projects. The project in California has been more about industrializing it and handing it over to a private lab to do the analysis. where here it's more of like kind of a grassroots, get people trained across the state in academic labs and you know, different types of labs. And so it's been fun to see like the pros and cons of both approaches. So yes, I don't know the website off the top of my head, but I can share it for sure.
I just did a quick Google and search, and I think I found it. But if you have a better link, I'm
I will
sure
share a link. Yes, great
others
link.
would appreciate that. Okay. So Jordan Sheet says, do you have any plans to look at SARS-CoV-2 variants?
Yes, in fact, we've started doing that, and both targeted and also sequencing. And we detect, it was interesting, we had some samples in California that we measured, that we went and looked for the British variant, B117. And we looked at several plants actually earlier from early and late in the fall. And we did detect it in one of the wastewaters much earlier than we were thinking we would. We saw it in mid-November, I think, in one of the wastewaters from the cities that we look at in California. And we thought, no way, this must be a mistake. Maybe our assay isn't quite right. But then we did some literature searching, and they've done some predictions, and they found that in communities in California, they do think that that variant was circulating as early November. And so if we verify that, you know, that shows that wastewater can tell you when these and then we detect the variants since, right? Especially now that they're becoming so much more prevalent. But even early, we were able to detect it in the wastewater. So yes, I think that's really exciting because, you know, as testing has ramped up for COVID, wastewater is still and it's become faster. Wastewater is still very valuable, but you compare it to how valuable it would have been early in the pandemic before testing, it's not as valuable as it was early in the pandemic. But variants, like, I don't know how quick we can detect every variant in every clinical sample we get, where using wastewater to kind of keep a pulse on how the variants are spreading through the community, we can develop an assay pretty quick and start incorporating that into our wastewater surveillance much quicker than we can get every clinical sample testing for these variants. So I think that's a really exciting area, and it is possible.
Well, it strikes me that, you know, it's a form of pooled sampling, and pooled sampling requires a low level of, you know, prevalence. And so if you're looking at when it finally arrives or something to appear, that's a great way to do it at a population level. That makes a lot of sense. I have another question from it's Laura and Larry on the chat here, and it relates to just generally the water treatment, the ability to treat wastewater and disinfect wastewater from COVID. So could you talk a little bit about the, you know, removal efficiency or how effective water treatment plants are at removing or disinfecting?
Yeah, sure. So in the case, so I want to just, it looks like, based on the literature that exists right now, that COVID does replicate in our gastrointestinal tract, but there was a really nice science, I think it was Science Translational Medicine article that came out last year that showed that if you take rotavirus, which is a common, you know, fecal-borne illness, and SARS-CoV-2, and you put them into, you know, the fluid in our stomachs, that SARS-CoV-2 is inactivated a lot faster than rotavirus. Rotavirus can persist in those gastric juices in our system, where SARS-CoV-2 appeared to lose its infectivity very quickly. And there have been attempts to culture SARS-CoV-2 from feces. There was some early on reports of successfully doing so, but I understand there have been many attempts since, and just it doesn't seem like the feces, even though they're loaded with RNA, they don't seem to be loaded with infectious viruses. That's not to say that it's not there, and we very well could hear of more cases where it's cultured from feces, but it doesn't seem like it's going to be at the levels of some of the fecal-borne illnesses or those viruses. But so, and that work that I had done back in 2016 was kind of hoping to get at that question: if there was a virus like SARS that was excreted in feces and did make it into feces in high levels, what would happen at the wastewater treatment plant? So first of all, they'd settle more. So we probably have a more we have an enrichment in the solids compared to the other viruses that we usually care about. But it's inactivated faster in wastewater. The coronaviruses are, and it's very susceptible to oxidants. So if we disinfect, and also UV, very susceptible to UV. So if we disinfect at the end of wastewater treatment, and it's been designed to take care of the enteric viruses. I feel confident that for SARS-CoV-2, we're doing an excellent job of inactivating it. So I think our system was designed for kind of the hardiest viruses and the highest concentration viruses that we have in our wastewater, and this tends to be just a wimpier virus.
So takeaway for both probably drinking water and wastewater, the risk of treated water having infective
Very, very,
COVID-2 is very, very low.
very, very low.
Okay. I'll open the floor up again for more questions, but I have to get a question in here because this is something that my wife would probably kick me in the shorts if I didn't ask before that. You know, you mentioned at the beginning of your talk that at the beginning of this pandemic, you know, we were one of the people that washed every grocery that we got, and all this stuff. And over time, that's kind of lapsed. And I'm just curious as to, based on your research and what you understand of this, as far as, I'm sure you're a proponent of mask wearing and whatnot, but general hygiene kind of things, washing countertops, ventilation. Can you give just a perspective on what you think is probably the one we should be paying attention to most when we go out in public?
Yeah, so I think and I think so much has come out of the epidemiologists. This is what they do. They figure out where are we getting the virus? What are the transmission routes? And so I think there's a difficulty in communicating because the majority of the transmission is happening through close contact or aerosols or droplets. Doesn't mean that it's not possible that it could be transmitted by fomites and surfaces, right? It's not driving transmission, that mechanism, but that doesn't mean it couldn't. If somebody, touches their face and touches a surface, and you follow right along and grab the rail right after they did and then touch your face, yes, transmission could happen. So hygiene is still really important. But I think when it's about reducing risks, right? We're engineers, we want to reduce risks. I think the bang for the buck comes from protecting what you're breathing in and keeping a distance, avoiding exposure from the small droplets and the aerosols that are being emitted from people. And so that's where when I'm in public and when I'm in a cramped space, I just want to get away as far away from people standing around me as possible to get that buffer, because I think that's what the epidemiology suggests. But that's not to say that we shouldn't still be careful about touching surfaces. I'm not, we don't disinfect our groceries anymore because the chances of somebody happening to touch that with COVID right before it gets dropped off at your house is very, very low. So yeah, for me, it's not worth the reduced risk of what we've gained from that.
Yeah, I was struck in one of your earlier slides where you talked about the percent humidity, relative humidity, and the temperatures that's required to inactivate viruses on the surface. And while you were using higher temperatures, the percent humidity, once it gets above 30%, which most households maintain a relative humidity of 30%, so like that's not that's a crazy high number.
Right.
So
but you can imagine that maybe in a really dry, cold climate, that's where you might see the virus.
Yeah, and that explains a lot about the winters, right? Where we struggle so bad in the winter. Great. All right. Time to turn the floor back over to others here. From Andre, we currently use RNA copy as a measurement unit to detect the presence of SARS-CoV-2 in wastewater, and there's no publication yet related to obtaining an infectious virus from raw sewage. Does it mean that the risk is low in terms of contact with raw sewage? And is there any model available for SARS-CoV-2 dose response yet?
Okay. So, right. So when we're doing, that's a good point. When we're doing this wastewater surveillance, this is all genetic material, right? We're measuring the RNA copies. It's not, has nothing to do with infectivity. I know that Chuck Gerba did have some work on culturing. He was trying to culture the virus from wastewater. I haven't heard if he's ever been successful. I assume that if they'd been successful, they would have announced that. So I haven't heard. I've heard of it in some, you know, rare cases from feces, but not from wastewater. So, you know, I think I will say that when you're working with wastewater, my team who works with it, especially when we enrich the solids, we are treating that. I think the CDC, their recommendations are you're treating it like a BSL-2 plus waste. And so we're kind of assuming that it might be there, but I suspect it's there. And if it's there and it's infective, it would be at really, really low levels. And I haven't heard of culturing. And then the second question was about dose response.
Yep.
For SARS-CoV-2, I've seen some for UV, and as expected, it's got a big genome, which makes it more susceptible to UV than most RNA viruses that we've tested before, so it's pretty wimpy. I'm sure they exist. I have to admit, I haven't kept up with it's hard to keep up, I'm sure you all know, with literature these months. There may be dose responses for chlorine and other treatments. But we do have them for the other coronaviruses, and there's nothing about SARS-CoV-2 structurally that would make me think that it would be so different from the other coronaviruses. And again, with chlorine, with UV, with other disinfectants, I think they tend to be more susceptible, so faster inactivation kinetics than norovirus or enteroviruses or things like that.
Right. Well, that's great. I think we're going to have to leave it there. We're out of time. So Krista, I really appreciate the presentation. It's really informative and certainly still very relevant research. So before I let everyone go here, I want to bring up that the next Water at Wayne seminar will be on April 8th, so a week from today. It'll be given by Dr. Andrew Gonwald. I hope I pronounced that right. And he'll be discussing Great Lakes water level variability and kind of what that means. A recording of this event will also be sent out after. It'll take a little bit of time for all the technology to kind of catch up here, but that will be sent out, I believe, to the listserv afterwards. So with that, I will thank everyone for joining, and well, virtually thank you, Krista, for a wonderful presentation.
Thank you. Thanks. Take care, everybody.
Thank you.
Thanks. Great job.
Thank you.